Hi everyone, my name is Jessica Solis, and this summer I had the privilege of working at UCSF Genomics CoLab with my mentors Arnab, Ghosh, and Eckalbar, in which we analyzed the impact of dexamethasone and interleukin-8 treatment on gene expressions associated with chronic obstructive pulmonary disorder, otherwise known as COPD. COPD, as we know it, is currently an incurable lung disease that causes airway issues such as emphysema and small airway fibrosis. We know it's caused by both genetic and environmental factors, and that COPD causes degeneration of bronchial smooth muscle cells. If you look at this diagram, in a normal, healthy bronchial tube, normal airway flow is not inhibited and is normal. However, in someone who has COPD, their bronchial tube is extremely inflamed by a cytokine known as interleukin-8, which causes this inflammation and inhibits normal airway flow. We do know that there is a potent anti-inflammatory corticosteroid known as dexamethasone that has been used to help treat respiratory disorders. Yet what we do not know is the actual transcriptomic landscape of genes that are associated with COPD when treated like corticosteroids, like dexamethasone. Thus led us to the question, what impact does dexamethasone have on smooth muscle airway cells when stimulated by interleukin-8? To go about answering this question, we began with cell culture of bronchial smooth muscle cells from 3 different biological replicates. We allowed them to grow to 90% confluency, and once that 90% confluency was reached, we serum starved them for 24 hours prior to treatment. After that 24 hours, we began treatment. As you can see here, this represents our experimental setup. We have our no treatment serving as our control, interleukin-8 only with a concentration of 100 nanograms per mil, and a combination of dexamethasone and interleukin-8, where dexamethasone had a concentration of 40 nanograms per mil. This treatment went on for 24 hours for all 3 biological replicates. And after that 24 hours, we then harvested 250,000 cells from each of the wells. Once we harvested the cells, we were able to extract RNA from them and then prepared 350 nanograms of cDNA for our qPCR. For our qPCR, we tested all of our biological replicates using technical triplicates and also using the delta-delta CT method for analysis. Additionally, we used actin B as our housekeeping gene for our qPCR and chose 3 specific genes that were strongly associated with COPD. As a result, you can see in our first gene of choice, elastin, that has to do with tissue repair, that we tested the relative fold exchange. In black, we have our no treatment, and in the center, we have our interleukin-8 treatment, and in the last column, we have our dexamethasone and interleukin-8 treatment. As you can see, when the cells were treated with interleukin-8, there's a pretty high gene expression in most of these cases for elastin. However, when the cells were treated with the combination of dexamethasone and interleukin-8, you can see that the gene expression reduces pretty significantly. We see the same trend occurring in our next gene that we tested, fibulin-5. Again, you can see the dexamethasone helps to reduce the inflammation caused by that interleukin-8. However, in this last case, for one of our biologic replicates, this showed the opposite trend. But again, we see the same trend happening in our last gene, MFAT4. The dexamethasone does a great job at reducing that inflammation and lowering the gene expression. And because of this consistent trend, we know that dexamethasone has the capability to significantly reduce the inflammation of the impacted airway smooth muscle cells, which can be beneficial to create therapies to better treat COPD. As for future directions, we intend to test more genes that are associated with COPD in a very similar way in qPCR analysis. And if that shows very promising data and a similar trend, then we'll move on to bulk RNA sequencing to get a more in-depth analysis there. I'd like to quickly thank Gladstone Institute's and Melanie Ott for creating PUMAS program, and of course, Emma Suda for coordinating it all. I'd also like to thank all of the Genomics CoLab and Eckalbar for allowing me to work in his lab for the past 8 weeks, along with my amazing mentor, Arnab Ghosh, for teaching me everything and being so patient while I learn all these amazing techniques. This wouldn't be possible without funding, so thank you so much to our sponsor. And of course, thank you to my community college and Melinda Kitts for recommending me to this PUMAS program. If there are any questions, please feel free to ask them now.
Wonderful, Jessica. Thank you for the presentation. Any questions for Jessica? Is serum starvation required for the effect of the dexamethasone? Is your question, Jessica?
So the reason we did that was basically to kind of prep the cells without any influence of anything else. So what we mean by serum starve is that we use the growth media without any growth factors in it. So it just kept the cells healthy, but it didn't allow them to grow past the 90% confluency. So get the full effect of the treatment for the